Krebs cycle, which is also known as the citric acid cycle, and also the TCA cycle. Those are the other names referring to basically the same thing. And lastly, the wonderful process known as the electron transport chain. So the glycolysis involves 10 stages, which we shall get into later on, but maybe as a matter of clarity, when you look at the word glycolysis, it is a fusion of two words, that is glyco and lysis. And initially, that was a long time ago, glucose was called glycogen. So that's where we get the word, the term glyco. And the term glyco simply means a sweet, simply means a monosaccharide, and simply means a 3-carbon molecule. And then lysis means breaking down. So maybe that's where we get the term where people simply call a respiration the breaking down of glucose into those other the constituencies that we know of. And then we move on to the Krebs cycle. The Krebs cycle basically involves 8 stages. And the ETC involves those complexes that I have explained. Okay, so let's look into our slides to see whether we've missed any other important details. So I'm just going to read out what I prepared on the slide. So we have looked into the site for respiration, which is our mitochondria right there by the corner, and the stages of respiration, which is glycolysis. We shall get to look into glycolysis as a topic, and the citric, which is also called Krebs cycle, the TCA cycle as a subject, and then one last stage which is missing there, which is the electron transport chain, which leads to the oxidative phosphorylation of our ADP to produce the ATP molecule that we want. And we have stated that mitochondria is an organ which is suspended inside the cytoplasm. It is the site for respiration. It is a double membrane organ. It has 2 membranes, that is the inner membrane folded into Christi, the intermembrane space, and the outer membrane. And okay, fine. This leads us to the issue of the ATP cycle. ATP cycle is a process which goes on because we always want ATP in our cells for the sake of cellular respiration, and the cellular respiration which is essential as far as cellular processes are coming. And I believe that we have tied on the issue of explaining how the important molecule, which is a high energy molecule, the adenosine triphosphate works, and we have explained that it has an adenosine group followed by 3 phosphate groups, and we have explained reactivity in terms of these 4 phosphate, these 3 phosphates, which are very reactive groups. And so basically when you talk of an ATP molecule, you are referring to a high energy molecule, and it is a short-term storage for energy, which you can call as an energy buffer, because you understand from computer science that a buffer is a temporary storage location. You don't store forever, you're just storing some important information momentarily, and then you get to use it when need be. And then from what we have learned and appreciated, we know that ATP is a highly reactive molecule, as denoted by the bond, which is a squick line, which is right there. It can easily be broken by hydrolysis, that is the addition of water. And that means that when we have added water to a substrate reacting together with ATP, and ATP for the sake of the powering of the whole process, what will happen is that due to hydrolysis, the substrate is going to gain a hydrogen cation and also a hydroxide that comes as a result of the addition of water, as a phosphate is liberated from our ATP, and what the resultant molecule from ATP is our ADP. And then we have come to a position whereby we appreciate that as our bonds are broken, what result is release of energy, and also that our ATP is a phosphorylated nucleotide. Nucleotide, maybe you are referring to things like the genetic material that is added, a phosphate group to it. And basically what we have there is the equation for respiration, and seeking to understand what is going on. Breaking of the bonds from glucose to water and carbon dioxide as low heat ATP is produced. So, like alluded to earlier on, whenever there is a breaking of a bond, heat is produced. When hydrogen and oxygen combines on the ETC, electron acceptor, heat is released. It is an exogenic reaction. Okay, and then lastly, is it lastly? Yeah. Lastly, we're going to understand the molecule glucose, since it is what the central molecule when it comes to the issue of respiration, because respiration is all about harvesting the energy, the energy from the fuel, and the fuel molecule is what we have as our glucose. And as animated by that diagram here, what we have is a glucose molecule, which we can call a monosaccharide, simply meaning a simple sugar. But now what we have to understand is also it is known as a hexose, and want to understand why it is so. It means that it is a 6 carbon molecule, and want to understand the arrangement of the carbons, because the arrangement of the carbons, as we shall get to understand, determines the 10 stages of glycolysis in terms of reactivity. Because what we want to have at the end of the day is a molecule which has got more energy for the sake of generation of our ATP that we want. So you'll understand that the first carbon is located here. So maybe I can just hexose, meaning 6. So here we are. This is our glucose molecule right here. So what we have here is our carbon number 1. This is carbon number 2. This is carbon number 3. This is carbon number 4. And this is carbon number 5. Then carbon number 6 right there, and this is our oxygen going that way. So you'll understand that as we move on, like as you shall get to realize from glycolysis, you realize that the first stage involves a substrate phosphorylation. That is when we add to our glucose molecule an inorganic phosphate so as to destabilize it. And then the second stage of glycolysis involves an isomerization reaction, whereby you are changing the configuration of the glucose molecule, also by opening and closing this ring, because what you want at the end of the day is to expose this first carbon molecule, which is sort of hidden, and when it is exposed, the final configuration of your first, the ring is opened, and it is somewhere here. What you have done is you have made the molecule more reactive and to be ready to get to the next stages of glycolysis. We shall get to learn more about that. So that being said, I'm just going to summarize by reading what is on the slide. You realize that a glucose molecule it is a monosaccharide, it is a hexose. Respiration can be aerobic or anaerobic. The goal is to produce energy so as to power the cellular processes, and glucose as it is a fuel for respiration. So earlier glucose was called glycose. This should explain the term glycolysis, glyco, sweet or monosaccharide, lysis referring to the destruction or the breaking down. So glycolysis is the first stage of, be it aerobic or anaerobic respiration, since it requires no oxygen, and you shall get to see how it is like that. And finally, what we have there is an animated diagram which summarizes the whole process of respiration. As you see, the product of glycolysis is what is known as a pyruvate, which are two molecules of pyruvate. And anaerobic respiration, what you have, it will enter into the cycle known as the Krebs cycle, and then what we have at the end of the day. But before it gets into the Krebs cycle, it has to undergo a stage which is known as decarboxylation, whereby we lose our carbon dioxide molecule as a way of preparing it to accept a 4-carbon molecule, oxaloacetate, that we need to react with it to form a citrate, which is the beginning of our citric acid, our Krebs cycle. And then if it is that it is anaerobic respiration, it can take two paths, that is alcohol fermentation, or this is anaerobic respiration in animals, whereby a product that is known as lactate, think of what you call that, lactic acid, which is a pain that you feel in the muscles due to insufficient oxygen. But when muscles are trying to generate energy that they need for the processes, the rigorous activities that they will be undergoing. And at the end of the day, what we have when glucose is done is a water molecule and carbon dioxide. When you get to this point, you know that you have terminated the whole process of respiration. So as you can see, these initial steps here, the pyruvate has got different fates. It depends with the pathway that it follows. When it is under anaerobic conditions, it either goes to ethyl alcohol formation, or it goes onto the lactate formation, which is what happens happens in humans, and this happens in plants. Because if you get alcohol in your muscles, you would die. It is toxic. It's a toxic acid. It is a toxic substance. But now, if the pyruvate undergoes reactions under aerobic respirations conditions, what you get is the desired outcome, which is your water, your carbon dioxide, and the ATP that you need for the powering of cellular processes. So as far as our introduction to the respiration is concerned, we have gotten to the end of it. Next time we meet, we shall be getting into the nitty-gritties pertaining to all the stages involved. Thank you.